So I'm gonna talk a little bit about one of the things of my heart, which is heart rot. So this is all based around some research I did in my master's. So this is going back about 5 years now, but I'm talking mostly about a paper that should hopefully be published in the near future. It's going through review at the moment, so fingers crossed. And it all starts in woodland. So ancient woodland is woodland which includes trees that are particularly old. And these old trees are often referred to as veteran trees. And they're basically defined by a load of different features, such as holes in their side called rot holes or hollows, which would be one of the main focuses of this talk, but also things like the crowns become slightly more developed, the roots buttress, and they're generally quite gnarly as trees go. And this is really, really beautiful woodland. So it's home to a lot of different animals and plants and even fungi, but sadly it's disappearing from the UK. So during the 20th century, we had several great conflicts, and great not as in good, but as in very large. So the world wars being the prominent examples. And during these, because of the war equipment and machines often being made primarily using wood, we actually chopped down a lot of our forests to basically fuel the war effort. So a lot of the trees that would be getting to this old stage over sort of 80 to 100 years where big beech trees become veterans, those trees are missing because we chopped them down for wood. So we have this thing called a generation gap where we have young trees, we have the very old trees dying off, but nothing to replace them. And that's really bad news for a whole load of different organisms, particularly those that rely on these small micro habitats, so little features on the trees that help. And I've mentioned briefly rot holes already. These are fantastic habitats for all sorts of different creatures. So that it all starts with fungi getting into the tree, or sometimes already being present in the tree, and basically decaying the heartwood. So you can see in this left image, we have all of this beautiful coloring, and you'll see various bowls and things like that made using this kind of wood, referred to as spalted wood. And this is basically where heart rot fungi are taking over. You can see these black lines actually where two different fungi are fighting. And eventually this sort of fungal colonization results in hollowing, where the wood begins to rot. And eventually it just becomes this soupy, gloopy sort of tree diarrhea, basically. And that's fantastic for some organisms, not particularly palatable to us. It absolutely stinks and it doesn't look particularly appetizing, but invertebrates absolutely love it. So you get all sorts of different invertebrates that depend on these hollows, including slugs and worms. You can see a few worms there. The worms actually climb the trees to get into the holes as well, which is a whole other story. We have things like featherwing beetles, which are some of the smallest beetles. They don't have many of their vital organs. They sacrifice them to be smaller, and they're basically hitchhikers on the wind using these feathers instead of wings. And this is the Batman fly. It's called the Batman fly because it has a logo on its back there, the black mark that looks a bit like the Batman logo. And particularly pivotal to this talk are click beetles. So there's one particular click beetle that lives in the UK that's of particular note. So the violet click beetle, Lemonuscus violaceus, lives exclusively in 3 sites in the UK. And it depends on these really specific habitats, these basal holes, so holes right at the bottom of the tree that lead into this almost like black plasticine-like wood mold. So it's a particularly sort of nutritious and foul-smelling rot, but these beetles absolutely love it. But there are only these 3 populations left, and it's thought it's because of the very specific habitat requirements they have. And obviously, when its habitat is continuing to disappear and there's nothing to replace it, we're starting to worry about this beetle, and we want to try and save it. And that's effectively where my work comes in. So during my master's, I had 3 aims. I was looking at the invertebrate communities, what they were actually comprised of, which species were there, and effectively how that changed between different rot holes. But most pivotally for today, I was looking at the habitat characteristics and the volatile profile of the violet click beetle habitat. So basically what kind of mold it likes to live in and how does that smell. So I went to some beautiful sites for my master's work. I went to Windsor Great Park on the left there. That's Windsor Forest. You can see a castle. You might be thinking, I'll just point out with the cursor just there. You might be thinking Windsor Castle. That's the Queen's house. No, that's actually Legoland. It was literally next door to Legoland. Fantastic. In the middle here, we have Epping Forest, which is just north of London. And on the right, we have Savernake Forest, which is deep in Wiltshire. So I basically managed to convince some people to let me go on little frolicking days out looking for beetles instead of doing real work, which is absolutely great by me. But that did involve basically sticking my hand into what is effectively tree diarrhea. So I don't know if you've seen Trainspotting, the scene where he goes into the toilet. That was basically the tree version of my life for a year. And I'd take this rot out of these holes, bring it back to the lab, and I basically picked out all of the different invertebrates. So I'd get things like millipedes and spiders and beetles. And then I'd put all of this material into these special things called Tolgren funnels, which are basically just a little cone that heats and dries things. So all the little invertebrates run away from the heat and they fall down to the cone where we can collect them. And that's where we've got a lot of the smaller invertebrates, things that I couldn't see with my own eyes, things like pseudoscorpions, which you can see here, or mites and all sorts of tiny things. So I also looked at the actual characteristics of the rot. So this was a very important part for the violet click beetles story particularly. So I used a potentiometer to look at water potential, which is basically the mobility of water, how much water is moving around. I measured the content of the water and the density of the mold and all sorts of things like the diameter of the tree and the height of the hole up the tree, just to try and look at what different things might be affecting the violet click beetle habitat preference. I did volatile organic compound analysis as well. That's a lot of long words. I used gas chromatography, mass spectrometry to do that. That's even more long words. It's basically just using big machines to work out how something smells. And I basically just did that with samples of rot from different trees. So looking at the actual habitat of the violet click beetle, we basically found that this is one example of such a habitat. The tree diameter was typically larger. So there are three categories of trees here other than those containing the violet click beetle. And these are based on surveys for the violet click beetle. And they basically looked for trees that they think might suit the habitat, the most kind of similar trees. So they would look at things like the diameter of the tree and the type of rot that's in the tree. And you can see that they're all quite high diameter. So a beech tree being about 80 centimeter diameter is pretty good. That's a pretty old tree. But the ones with the violet click beetle in are just that little bit wider. And things like the density differed a little bit from some of the other categories. But the only thing that actually significantly differed between these categories was the water content. And actually, it's just the trees that were considered high potential for containing the beetle that were different, not the actual trees containing the beetle, which isn't particularly helpful, but it's interesting nonetheless. And this is a slightly harder to explain figure, but each of these points is basically a different tree, and they're separated based on how differently they smell. So the further apart the points are, the more differently they smell. The blue ones are all in April, with the darker ones being the trees confirmed to have the beetle in them. And as it gets lighter, the lower potential for the beetle to be there. Gray is the same thing, but for May, and red is June. So you can basically see here that the trees smell very different in April. But as soon as you get into May and June, they start to smell a little bit more similar. But always we're getting the actual trees containing the violet click beetle coming out slightly differently. So they do have their own distinct odor, which is very interesting. And then looking at that kind of disassembled into just the month or just the tree status, you can see there's a little bit more overlap, except in the months there's quite a big difference. So that was very interesting. So you might be wondering, what does this all do? How can this help? Well, the fact that we found these different smelling trees, it basically helps us to find odors that we might be able to use to attract the beetle. So we might be able to bring all of those beetles into one place, to either count them to see how many are actually left, so we can base our conservation plans around that, or we can actually use it to attract them to potential new habitat that maybe they wouldn't find otherwise. to try and give them a better standing chance of actually surviving. We also got a good idea of the habitat characteristics of the beetle. So we can use this information to basically help us survey for the beetle. So whilst these surveys are already seemingly doing a pretty good job, we now have quantitative characteristics to look for, which is a little bit easier in some ways. But ultimately, the whole aim of this really is to just allow the beetle to reproduce. So hopefully we'll have a thriving population before long. There were some questions left unanswered though. So obviously the decay to create these rot holes, as I mentioned at the start, is all just to do with fungi. And there's a really dynamic community of fungi present in all of these trees. So to actually look at the fungi and whether the beetle is relying on specific fungi is a whole other question, and one that I think would be very interesting. We tried to look at this a little bit, but it didn't go particularly well because we didn't really have the tools available during this project to do so. So that's something that hopefully someone else will look at eventually. But also these trees were only looked at in 3 months. So we looked at them between April, May and June, as you saw in the smell profiles. And it seems that these are very dynamic habitats. There's a lot of community turnover, which means the species change quite regularly. You have spiders moving in 1 week and then the next it'll be a different beetle. So looking at whether the differences over time affect its suitability for the violet-click beetle is another really interesting question. Can they become unsuitable after a while? Who knows? Tune in next time. But ultimately, the big question is whether these volatiles that we've identified, the smelly compounds, whether they can actually be used to do this surveying for the violet-click beetle. So we need to look into whether the beetle is attracted to the pheromones that we can then artificially reproduce. But like I say, a question for another time. But thank you very much for listening. And thank you to i3 for having me. And thanks to my lab group at the time. This is a slightly old picture. I think Yoda's aged a bit since then. And thanks to Bug Life for partly funding my project. Any questions?
So yeah.
Thank you very much, Jordan, for such an interesting talk. I feel like we have to slow down a little bit because with this new YouTube, we obviously have a delay. Something good to know. So yeah, so if anyone here has any questions and they'd like to kick off with a question and then see if we get any other coming through the YouTube or the Facebook. But yeah, if anyone has any questions, please go ahead. No. If not, I have one. I do have one. So if you've only looked at beech trees, do you know if, for example, that these beetles inhabit other trees, or basically, if there's less beech trees, do we know if these beetles have found a new habitat where they're starting to thrive? Have they found an alternative place to live?
So, very good question, actually. So actually, I did only look at beech trees for what I've shown you here, but the 3 sites that the violet-click beetle is present in, I misleadingly told you about my field work in Epping Forest and Savernake Forest, which has actually nothing to do with the violet-click beetle. The 3 sites are actually also Bredon Hill and Dixton Wood. And Bredon Hill is a population that's entirely present in ash trees, so a completely different species of tree. But I did actually look at the violet-click beetle trees over there as well, using volatile analysis and all the same different tools. But at the time, we didn't know exactly which trees would contain the beetle. So it was a little bit blue skiesy. And because of that, the actual results can't really be used to pick apart any actual patterns. But it's definitely a really interesting question and one that I think definitely needs further investigation.